So in this session, we are going to look at the final part of the evaluation and conclusion of your paper 5 of an experiment. So we've looked at the data, handling of the data already. Now we're going to look at, so for the second part of paper 5 is handling of the data, and then an analysis of the data, then evaluation and conclusion. So now we're going to look at evaluation followed by conclusion. Okay. So evaluate the data and experimental plan means after the lab assistant or you yourself have done and collected the data, you want to look at your data as well as the experimental plan or design to know whether the results that you have obtained is valid, it's accurate, and is reliable or not. Means precise or not. 3 things: valid, accurate, reliable. Valid means whatever you measure can show what you want to find out. Like I measure the number of bubbles released. Can the number of bubbles released per unit time represent the rate of respiration? So whatever you've chosen to measure, does it really represent what you want to measure or not? What you want to know? That's called valid. Okay. If I want to know the mean height of the male students, I cannot be measure everybody. Okay, I cannot be measure the teachers, the lab assistants, okay, the admin staff, all the female and male, and then I use it as the mean height of the students, male students, right? So this is called validity of your experimental plan and data. Okay, accuracy and reliability, you're already familiar, so I'm not going to talk about it. So this is where you want to evaluate your data and experimental plan to see whether your result is valid, accurate, reliable. If your results they are valid, accurate, and reliable, then you can proceed to do conclusion. Okay, right. So what are the things that you will look at is number 1, you want to look at the anomaly in your experiment. So we need to identify them in a table or graph. Then we need to suggest a possible cause of the anomalies. Is it because the wrong solution is added in, or is it because suddenly there's an electricity cut and then the water bath is no longer hot, say for example, or is it because you mix up the solution, okay, or you mix up the procedure, okay? So what is the possible cause of the anomaly? And suggest strategies for dealing with such anomalies. So usually when you see anomalies in your result, we will try to repeat the experiment to get more readings, okay? If you're in the lab, you can still repeat experiment, okay? Or you can still go to the lab, you can still repeat experiment. But once you've completed the experiment, you are no longer in the lab or you're out of the field already, you don't want to repeat experiment, you omit the anomalies. Omit the anomalies from all the calculations that you want to do from then onwards, right? Data analysis, right? Calculations. So calculation of mean, median, mode, calculation of all the statistical analysis, you omit the anomalies. Because anomalies are all error, they add error to your calculations. Your calculations will not be accurate, reliable, okay? You can also show where are the location of the anomaly in your graph and exclude it from drawing your best fit curve. Okay, say for example, this is your graph. Okay, okay. So you have got a point here. Here, you know it's a straight point, huh? Right, so then you can see that actually this looks like an outlier, means the anomaly. So you may want to circle it and label it as an anomaly. And then you draw the best fit curve. Best fit curve is quite difficult here. You need more points. You need one more point, right? Okay. say for example, you got 1, 2, 3, 4, 5. One more point here. So this is also an anomaly. Straight line. Okay, doesn't look good. Huh? Okay, this anomaly must be further away. Okay, so the distance of this anomaly from the best fit curve must be the same as the distance of this anomaly on top of the curve. Top and bottom must be same distance. Then that's the best fit curve. So these two are anomaly. Okay? Right. After identifying anomaly, you may want to see the adequacy of the range of independent variables. So what do you mean by adequacy of the range of independent variable is, okay, I've done the experiment, I've got the result already. I find out that I cannot find where is my optimum temperature. Okay, I've done my experiment on this enzyme, starting from, let's say, 0 to 60 degrees Celsius. And what I can find in my graph after I plot my graph is my reading going up. It didn't turn down, even though it's 60 degrees Celsius. That means I cannot find my optimum temperature. So after that, what will I do? Oh, okay. So the range of independent variable that I've used is not enough for me to find out where's my optimum temperature. I must go and conduct a second experiment where I will use a few more temperatures higher than the 60 degrees Celsius. All right, so that I can continue to obtain data. Then I suddenly realize that, hey, okay, I've got a reading here, here, here. So then after that, I will know my optimum temperature is this one here. Okay, so that means my original range of data here is insufficient for me. It's insufficient for me to find out where is my optimum temperature. That could be a reason I have to repeat experiment. Okay, so my reading is at lower range or higher range, so I have to increase it. Now, second thing, I want to find out where's my turning point. Okay, say for example, I also do the same experiment, and my reading is this one. So if my reading is like this, right, I cannot know exactly where is my optimum temperature. It can be anywhere between here and here, right? But exactly where, I don't know. So after the first experiment, I get my data already. I have to repeat experiment again. I do my second experiment, whereby I will increase the number of different temperatures within these two temperatures, right? So adequacy of range of independent variables. So now repeat it with maybe 5 readings, 5 temperatures within this range. So I've got 5 more points to plot. Okay, let's say 1, 2, 3, 4, 5. Then I can find out my optimum temperature more accurately, isn't it? So that is the meaning of adequacy of range of independent variables. So take the data, check, okay, my independent variable range, is it sufficient for me to get the result that I want, to give me the information that I want, okay? Then I also must check the experimental plan, are all the standardized variables being considered, and are they all being controlled well or not, okay? So I must check the experimental plan of this experiment. Okay, temperature control, pH control already, volume, concentration control, the mass of organism being controlled, wait time all are controlled. Okay, so this you have to check, and how they're being controlled. Okay, so the next thing you want to look at is the confidence in which conclusion can be drawn. Depends on whether, so when you draw a conclusion, you always draw based on your results, isn't it? So how accurate, how valid, accurate, and reliable is your conclusion, right? How confident are you that your conclusion, confidence, how confident are you that your conclusion is valid, accurate, and reliable? it actually depends on whether your result is valid, accurate, and reliable, which actually depends on your experimental plan, right? Because your results comes from an experiment, right? So how confident is your conclusion depends on your result. And how accurate is your result depends on your experimental plan or design. So therefore, when you want to know how confident you can make conclusion, you have to go back to check your result, accurate, reliable or not. That means you have to go back to check your experimental plan, the whole plan. Can it allow you to get accurate and reliable result? Okay, so what must you look at when you want to know whether you are confident with your conclusion or not? First, you must look at the quality of the planning. Is it good or not? Go through every item in your experimental plan. So for independent variable, do I have minimum 5 values? And the independent variable that I have selected, does it accurately represent what I want to test or not? Okay. Say for example, you want to find out what is the metabolic rate of the shrimp, this particular species of shrimp. Oh, okay. Yeah, a particular species of shrimp, okay? So that means to say you actually have to go and find out what is the metabolic rate of this shrimp at different sea concentration of salt water. What is the metabolic rate of the shrimp at different concentration of salt water? So your independent variable must be salt water, that means sodium chloride solution, and none other salt. That means you cannot go to the sea and take the sea water. Okay, because different sea water got different sodium ion concentration and got different other ions. So you must use pure sodium chloride. So does the independent variable that you've chosen represent what you want to test as close as possible? Okay, so it must be valid. If you say seawater, then you must say which seawater. Okay, which part is it? Is it a seawater of around the Pulau Ketam, right? Seawater around Pulau Ketam, and which side of Pulau Ketam? Is it southeast, northwest? So must be very, very accurately described, or else it's not valid already. Okay, because some other people cannot replicate your results because of that. Okay, and also you may need to also talk about which timing, which season, which day, daytime, nighttime, all these things must be considered already. Okay, so comparable or not, suitable range or not. Suitable range, just now you already know lah, suitable range for you to get what you want, or optimum temperature. Comparable or not means, let's say for example, can I compare shape and color? I cannot compare shape and color, right? There are two different things, right? If I want to compare shape, my independent variable is different shapes, isn't it? Whereas color is different color. So you cannot take shape and compare with color. That's called comparable or not. So the independent variable that you've chosen, the 5 independent variable, it must be comparable. Okay? Now the next thing is dependent variable. Does the dependent variable that I measure, dependent variable is something you measure, right? Does it represent what I want to know? So for example, number of bubbles released per unit time, does it represent the rate of respiration? Time taken for methylene blue to disappear, does it represent the rate of photosynthesis? Okay, the distance moved, the distance of the moved by the water in the photometer, does it represent total respiration, total water transpired? Okay, so you need to know how accurate is your dependent variable representing what you want to measure. Okay, any assumptions, right, will actually reduce the accuracy already. Okay, because you assume the shape of the leaf is round, but actually it's not round. Okay, then you calculate using pi r squared. Every assumption introduces some error. All right, assume all the water taken up is actually transpired, which is not true because it can be used for photosynthesis, turgor pressure, okay, hydrolysis. So assumption introduce error. All standardized variable considered or not, well controlled or not, okay? Procedure follow accurately, not step 1, add this volume to that volume, step 2, heat it up for 5 minutes, step 3, cool it down, step 4, neutralize. So procedure follow correctly or not in sequence, okay? Accuracy and precision, so do you have sufficient replicates, at least 3 readings? Do you calculate the mean or not? Do you allow time to equilibrate or not? Okay. What kind of instrument you use to do the measurement of, let's say, light intensity, color intensity, pH, what do you use to measure, right? pH meter is more accurate than pH indicator, okay? Or pH paper. Do you do a control experiment to eliminate possible uncertainty of the result? Because there are 2 factors which may affect your result. Have you done your control experiment to phase out, to find out, is it 2 factors also affect the result or only one of them? So is your result accurate or overestimation or underestimation? So you have to do control experiment. Okay, so after having considered the quality of your planning is good, then we know the result you obtain is accurate and reliable. And is also valid. Okay, then your conclusion, you are confident that your conclusion is also valid, accurate, reliable. Okay, so this is the first thing you want to check before you want to do conclusion, to make sure your conclusion, you are confident with your conclusion. So the first thing to check is your planning of experiment, every single part of the planning. And the second thing you want to do is you want to do the statistical analysis. Okay, statistical analysis will show you whether the differences between the sample is significant or not. Like just now I showed you, you compare the mean of height of student in Taylor's versus the mean height of student in Sunway. You say that Sunway student is taller than Taylor's. This is not accurate, reliable, and valid, because this is just a calculation from every individual. Every individual, the range of the height is so big. So if you take the whole range and you compare, then it is not significantly different already. So you need to do a statistical analysis to verify whether the differences between the mean of the two samples are significantly different or not, by considering the range of the data or the range of the mean that you have collected. Okay, so therefore, these are the 2 items that you need to consider in order to know whether your conclusion is valid, reliable, and accurate. So then you're confident with the accuracy, reliability, and validity of your conclusion. Okay. Now the last part, of course, is conclusion. All right. How to make conclusion is, okay, I've got this data, I've done all the analysis and evaluation already. I'm ready to do my conclusion. So how to do conclusion? You must always refer to the hypothesis first, as well as your result. Compare your result with the hypothesis, or compare your hypothesis with your result. Okay? So do the result or data support or negate the hypothesis? Then you quote the figures for the range of values that support and those that negate. So sometimes, let's say this is your hypothesis. When temperature increases, the rate of reaction increases. So this is your hypothesis, right? But when you get your result, you may get something like this, or you may get something like this, okay? Or you may get something like this. Case A, case B, case C. So for case A, then you can straight away conclude that, okay, so the results support the hypothesis. Therefore, as temperature increases, the rate of reaction increases, full stop. Okay? But if your result is case B, part of your data support the hypothesis, part of your data negate the hypothesis. So therefore, you need to say that, okay, for the temperature between this temperature to this temperature here, the data support the hypothesis, all right? But for the temperature T2 to T3, the data negate the hypothesis. So when the temperature increases from T2 to T3, you describe how it looks like. The rate of reaction remain constant, okay? At a particular reading, then you give the reading. So as the temperature increase from T2 to T3, you give the reading, then the rate of reaction remain constant at this reading, Y3, for example. So that is an additional statement you have to give, okay, to match the expected result. Okay, so for this temperature T1 to T2, as the temperature increases, the rate of reaction increases. So it supports the. So you can say that your results actually partially support the hypothesis. So for case B, you can say that the result partially support hypothesis. Then you tell me which temperature support, which range of temperature does not support. So for temperature T1 to T2, as the temperature increases, the rate of reaction also increases from what value to what value. Okay, Y1 to Y2. Okay, then for temperature T2 to T3, as the temperature increases, the rate of reaction remain constant at Y3. Okay, so that's what you need to write for your conclusion. So this is what I have mentioned just now about writing conclusion. Your hypothesis is when temperature increases, the rate of reaction increases. And the result shows that the rate of reaction increases until optimum temperature and then decreases. So this is another thing, right? So your results show this. Your hypothesis is this one. So what is your conclusion? The data partially support the hypothesis. Which part support, which part does not support? If it does not support, how does it look like? Okay, so the data or result only support hypothesis from this temperature to this temperature. Okay, T1 to T2. So write down T1 to T2. Okay, then the rate of reaction drops when the temperature increases from T2 to T3. Okay, so the rate of reaction drops, okay, from Y2 to Y3 when the temperature increases from T1 to T2. All right. So when the temperature increases from T1 to T2, the rate also increases from R1 to R2. Say for example, okay, this one is R2 to R3. Can use Y, can use R, can use anything, any symbol to represent. Okay, so that is how you do conclusion. So I have, let me recap what I've done for today. Yep. Okay, so planning is, okay, so planning we have covered last week already, right? Which involved these 8 steps in planning. So this is 50% of paper 5. So for the second 50% of the paper 5, it includes analysis, where you need to deal with data. You need to do calculations, recording of data, and statistical analysis. For statistical analysis, we can look at the spread of the data, which I've done it today. So we look at range, interquartile range, standard deviation, standard error, as well as error bar. Then, of course, in the next other lessons, we're going to learn statistical analysis for the differences between samples, whether they're significantly different or not. Okay, and there's one more thing I want to add in C is actually the correlation. Are there any correlation between the samples? Correlation between samples. So please add this into your notes. Okay, you're going to learn this in future. Then we also look at evaluation of your entire evaluation of your data and as well as your experiment, okay? So you want to look at your anomalies of the data, adequacy of range of your experiment, your standardized variable, well standardized or not, and also confidence of doing conclusion based on number one, the quality of the planning, number two, okay? So this depends on the quality of the experimental plan. Okay, accuracy, precision depends on quality of experimental plan, and also you also want to look at statistical analysis. Okay, so these two will give you the confidence of conclusion, whether conclusion is valid, reliable, or accurate or not. Valid means you're measuring what you want to measure. You're showing what you want to know. You're measuring what you want to know. Okay, valid. Measuring, mastering, measuring. What? It's measuring, measuring. How come measuring like that? Measuring what you want to know. Okay? Valid means are you measuring what you want to know or not? Okay? Accuracy, precision. Right? Now conclusion you have to look at. So compare your result with hypothesis. Okay? Sometimes you'll be asked to explain your result using theory that you've learned. How come the temperature is like this one? How come it changes this way? Okay? And then using your result to predict if temperature is this, then what should be the, if temperature is this, then what should be the result? Okay? Then any possible improvements that can be made to the experimental plan so that your results becomes more valid, accurate, and reliable. All right? So with that, we have actually completed the lesson for today. So now you're actually ready to try out a lot of questions so become more and more familiar with what I've presented to you today. So your homework that you need to do and pass up by Monday will be the second part of your practical notes here, right? Where's the second part? Okay. Second part here, right? Scroll all the way down after the notes finish. You see the sample questions here. So you need to do all the question except for the last question. So question one, you can revise again what is planning questions. Okay, and hypothesis, write hypothesis. Okay, then dealing with data. So this part here, what will be asked to deal with data? See here, they ask you to plot the histogram. See here, histogram, plot histogram. See, okay, and then you'll be given a table to fill in the blanks and do some calculations, and you'll be asked to do statistical analysis. Can you see there's an S and an SM here? So you need to know how to calculate, and you need to know what does it mean to you, okay? And then this one also calculate the standard deviation and standard error of the sample. So you can do this one as well. And then plot the graph, okay? So all the way until this question, you stop at question 2. Question 3 is chi-square test. It's a different type of statistical test that you have not learned yet. So we reserve this for the next time. Okay. Only next week I'll give you the evaluation and conclusion question because I think this number of questions already enough, right? Any one of you want me to give you more questions to try out the evaluation and conclusion? Or you prefer it next week? I think you do it next week better because you want to do a lot at one time. So I'll give you the evaluation and conclusion question next week to do, which is not here. I'll add it in again to your notes. You will see that I'll keep on editing and improving, adding more things to the notes to make it more and more complete. All right, so that you don't have to refer here and refer there, hopefully. So as I add more and more, you can go and print out those pages or you can copy out if it's a small section from the new notes that I have posted into your Dropbox, okay? So that's all for today. I hope that this session I've explained it clearly so that it's easy for you to understand and to master it and then to answer the passive questions. So that's all for today, okay? All right, I'll see you in the next session.